in the genetic class, in the general genetics, mutations can be contributed by two different ways, right? One is the big mutation, one is a smaller one. If you recall, one is the chromosomal mutation, correct? Another one is a DNA mutation. Chromosomal mutation, sometimes it is not inherited because it was terminated. Okay, for example, in mammals, if chromosomal mutation occur in which the chromosome are truncated or cut, okay, during the formation of the sperm and eggs. So when that particular sperm that has a truncated chromosome, when they are fertilized with the egg, the gamete is not viable, so it is terminated. Okay, so in some other instances, the mutated chromosome can be fertilized, so it will produce a fertilized cells, okay, zygotes and so on, and go on to produce individual with a mutated chromosome, but that one is quite rare. Okay, the normal mutation that you can see usually inherited is a mutation that occur on the DNA. Okay, so that is a DNA mutation rather than a chromosomal mutation. Can you figure the difference or not between these two? Okay, so that is the difference between chromosomal mutation and DNA mutation. All right. So in this lecture, we are dealing mainly with the with the DNA mutation. And so these are subject and tool of genetic analysis as well. So when we do mutation, in which we only change certain sequence on the DNA, usually it can be inherited and it can produce gamete and new organism, new individual with the mutation. Okay, so mutation that is important is a mutation that is heritable. Okay, a few of you mentioned don't forget to record the class. I think recording is
Record
in
the class already.
yeah, recording is already in progress. Thank you very much for the reminder. And Doctor, I just see gray screen. So, but now everything is okay, isn't it?
Yes, it's okay.
All right, good. Okay, coming back to this mutation that is related to genetic, usually a heritable mutation, meaning what? Meaning it is passed from one generation to the next. So that is what is meant by heritable changes. You know, sometimes in one of my, because I teach one of the lecture for genetic, general genetic, sometimes I mention you can you can get mutation when you are exposed to sunlight. You go to the beach, you know, something like that. When UV, you are exposed to the UV, then you are mutated. You can have mutation occurring on your cell. Okay, and if you are exposed to UV, very strong UV light, or sometimes you work at the facilities that use radioactive, you can also expose to this mutagen, the X-ray, the gamma ray, and so on. But let's say you are exposed, you are wearing a protective clothing, only you somehow forgot to wear a glove, or you are exposed some area. Even when you are wearing glove, the area that is the skin that's exposed can also have mutation, and it can, for example, develop into a cancer. That mutation is not a heritable mutation. Okay, so that is also a mutation but not heritable. But mutation that we are talking in genetic is the one that can be passed to the next generation. So that That is why it's called heritable changes. And this statement, mutations that are heritable changes in base sequence that modify the information content of the DNA is the one that is carried from one generation to the next. And you can have mutation that occur even without you exposing yourself to any mutagen. Okay, that one is a natural. When you have that, you can have a natural mutation, which almost every one of us have some kind of mutations earlier from our grandparents, our great-great-grandparents, and so on. Those mutations make us to be different from one individual to another can also happen. So that one is the one that can cause genetic variation among individuals. Induced mutation is the one where you are exposed. You are exposed to any agents that can cause mutation. So that is why here it is said, it's mentioned random mutagen and also specific target. Certain mutagen cause mutation randomly, and certain mutation can target specifically. Okay, all right. So these are example of how mutation can occur even without any mutagen. Okay, so you can see random mutation can occur by mutagen and also without mutagen. So for example, this one is a repetitive sequence of CAG. All right, and you can you can imagine that our DNA sometimes is fluidic. Okay, or even though they are wound around the nucleosome. If you remember the yo-yo line, the yo-yo of the DNA around the nucleosome. So sometimes these are open up and then packed again. So during this event, sometimes mutation can occur when some of the DNA are temporarily become single-stranded and then reattach. It will reattach again to become a double-stranded, but some of them are left out. So this one will change the length of the DNA. So this one is also a mutation. Okay, and then classification of mutation by effect of the effect on the DNA molecule. Okay, so there are several different ways of how mutation can occur. Substitution, you just imagine like in a game where you replace one player with another. So in this case, the substitution is replacement of one nucleotide with another nucleotide. All right. For example, purine to another purine, then it is a transversion when purine is swapped with pyrimidine. So that is a two different ways of substitution can occur: transition and transversion. You remember adenine, thymine, purine, pyrimidine. Adenine is purine, thymine is pyrimidine. Guanine is purine, cytosine is pyrimidine. So if you have a sequence of adenine, thymine change to guanine, cytosine, then is from A to become G. So that is a transition. Purine A change to purine G. Okay, AT become GC. So A change to G. So A is purine, G is purine. That is the transition. Whereas transversion, purine change to pyrimidine for that particular nucleotide. Okay. For example, in this case, you can see this one TA change to AT. T is pyrimidine, A is purine. So that is a transversion. Okay, and you can also have mutation by inversion, in which this one, not the nucleotide itself is swapped, but it is the inversion, a very short fragment of the DNA. So that one is inversion. And reciprocal translocation is the translocation means it is moved from one place to another, translocate. Okay, so parts of non-homologous chromosome change place, but how they change is because the sequence among those non-homologous chromosome are quite similar, so they can somehow temporarily align. Okay, for example, uh, you can have chromosome 1 and chromosome number 10. Okay, but somehow one of the region they have quite similar sequence. They can temporarily be side by side and they swap places. So that one is a non-homologous chromosome. And then you can also have chromosomal rearrangement in which one segment moves to another part of another chromosome. Okay, so you can have different type of uh, changes that can occur. Okay, chromosomal rearrangement affect many genes at one time because, as I mentioned before, in the chromosome, if it happen to have genes on it, they can probably have many different genes on that particular segment. So that will affect many genes. All right. So you can have inversion, you can have reciprocal translocation in which chromosome breaks and reattach at another place. Okay. Any questions so far? Okay. From the chat box, what I can see mostly, most of you mentioned only on the attendance. Okay, I will do the attendance later on. Okay, anything you want to ask? Okay, Nur,
Doctor,
yes,
for
Narissa.
example, yeah, hi. I want to ask, for example,
Mm,
if let's say a person is exposed to UV
yes,
and then they get, let's say, skin cancer, right?
yeah.
Can their, how do I say, is their mutation considered heritable for their children?
Um, okay, that kind of cancer, for example. Hold on, Narissa. Nurhana, you mentioned your friend cannot enter. Can she or he enter now? Because I haven't set any restriction. Any of you who has the link can enter. Ask your friend to uh, quit the WebEx and then rejoin it. Okay, rejoin WebEx. Okay, cancer, skin cancer, for example. You probably you read quite often in which those who like to uh, have their skin tan, okay, and they go to a tanning salon and they have a mutation that occur on their skin. So usually that one is non-heritable because it is specifically occurring on the cell on our organ, which is the skin, and that cell mutated, causing the cancer, and it will grow. Okay, that's the cancerous cell will grow. It will be localized because of the mutation that occur on a cell on the skin. Okay, but uh, some other cancer, okay, is heritable because changes in certain genes. So that one is also uh, uh, can also trigger cancer, but that is different from the cancer that is localized, like the skin cancer, for example. So that is, I hope I answered that, Narisa, and Bonnie also asked for
Yes,
mutation that.
thank you.
Okay, welcome. For mutation that cause cancer in our body, it is heritable or occur naturally or induced. It depends. As I mentioned, when I answer Narisa's question, it depends on cancer, because everyone carries certain type of genes. Okay, and some genes, when they are mutated, uh, and that mutation may cause cancer if it is later on triggered by something else. Okay, and a person might carry a cancer gene, but it is not triggered. Can also happen. So that one is heritable. So to answer Bonnie's question, is it heritable or occur naturally or induced? Usually cancer is induced. Okay, but cancer can be heritable. Okay, but cancer can also be can occur because you are exposed to certain mutation, but that cancer is not due to is not inherited from previous generation. Okay, so it can occur both ways. All right. Okay. Okay, we move on. Spontaneous mutation influencing phenotype occur at a very low rate. If you can still recall, in the general genetic class, mutation is measured in how many times it can be observed uh, in terms of how many thousands of the number. Uh, for example, like you can see over here, mutation rate is 11.2 mutation on average for every 10 to the minus 6, 1 million, for example. Right. So that is a mutation rate per number of samples that are investigated. So this is one way to measure the mutation. So 11 for every uh, 22 million. Okay, sorry, 25 mutation for every 2.2 million, which you divide this, you can get 11.2 times 10 to the minus of 6. So 11 mutation for every 1 million. So that is one way to measure mutation. So based on the number of samples that was observed. Another way to measure the mutation is by looking per generation. Okay, so that is also another way to measure the mutation. And you can see in whichever way that you check the mutation, heritable mutation usually, you can see the number is very low. Okay, why is it low? Where if when I say that mutation can occur all the time, the simple answer is that we also have a mechanism to repair the mutation, right? So that is why, although mutation can occur quite quite easily, it can be repaired by our system. So mutation rates from wild type to recessive allows for 5 code color genes in mice. So this one is a code color, this one is the number. So on average of this, so very low mutation rate. Okay, because it can be repaired. So that is why you can see the mutation rate is low. All right, the next slide. Natural processes change information stored in the DNA. So this one is how the mutation can occur because of certain mechanism. Okay, hydrolysis of purine base A or G occurs 1,000 times an hour. You can see this 1,000 times an hour in every cell. Hydrolysis of purine base, the purine itself, not the DNA. Okay, on the nucleotide. The so you recall that nucleotide arrangement like this. You can have hydrolysis of a purine base. Purine means what? Adenine and guanine, right? So for example, like this one, you can have a purinic site. There's no, you have phosphate group, you have ribose sugar, but no, what do you call that? No nitrogenous base. Okay, so you can see this one, deamination, depurination, you remove the nucleotide. Deamination, you remove part of the chemical group on the nitrogenous base. So deamination. Depurination, you remove the purine base. Deamination, you remove the amine group. So amine group, you have to recall amine, amino acid, right? And then when you learn biochemistry, the protein starts with the N terminus and with the C terminus. The N terminus because it has the nitrogen, and the nitrogen is usually on the amino group. So amino group is you remove the deamination. You remove this amino group, it become a hydroxyl over here. And you can see that the nitrogenous base from cytosine now behave as a uracil. So from cytosine, it become uracil, and you know cytosine pair with what? Guanine. Okay, I hope everyone guessed it correctly. Cytosine pair with guanine. But when the amino group is removed, it behaves now and it become a uracil. Okay, so when it become a uracil, you imagine this DNA that from cytosine become a uracil, it is undergoing replication. So when replication to uh, double-stranded DNA, it becomes single-stranded, isn't it? So this one contain cytosine that have been aminated, deaminated, become uracil. So this uracil now what will attach to it if there is a replication is occurring now? When it comes to uracil, will be bound with what? Adenine, right? So you now imagine the new strand having uracil and adenine over here. Okay, and this, okay, now imagine that this is adenine. So next round of replication, new DNA will, a new nucleotide will come, then this adenine will bind to thymine, isn't it? So originally it was CG, now it become AT. So now you have a mutation already, isn't it? For that particular nucleotide base. So this one original sequence, when you have the amination, uracil, and then it undergo replication, then the uracil will bind with a new adenine, and if this one undergo next round of replication, you can see that it originally from the two previous nucleotide, it is a CG, now it becomes TA. So now there is a mutation at this one. Okay, and if you imagine this code for an amino acid, now it become, let's say this one is CCC, now it become TCC, for example. So new codon, which represent new amino acid, which then change the sequence of the protein. All right. So that is how the process of mutation can occur and can cause changes, okay, in the cell. Doctor, in DNA mutation, does it occur in RNA as well? RNA, it is copying base. Okay, if this one is DNA, temporarily RNA is being synthesized. Sometimes it may have insert a wrong polymerase, RNA polymerase. It may insert a wrong RNA nucleotide. Okay, it can occur. Okay, but usually when we talk about this mutation, because we are talking about mutation that is inherited, isn't it? RNA is not inherited. it is wrongly inserted, it is wrongly translated, it will only affect that particular individual. But this one we are discussing on a mutation that can be passed to the next generation. Okay, mutation can occur during the transcription as well. All right, but then over here we are talking about mutation that is being passed from one generation to the next. Okay, all right. X-ray break the DNA backbone. All right, so that is why when you go for X-ray, uh, nowadays is quite safe, but you don't go because they have already studied what is the dosage that do not affect the cells. Okay, so the X-ray that you you will undergo when you go to the hospital for your medical checkup or whatever, you do not need to worry because they have already done study at what dosage, the timing of exposure that will not affect the cell and cause mutation, right? So you do not worry about that. But even for, when mention X-ray over here, there are different strengths of the DNA. Different strengths of the X-ray itself. Okay, so certain strong X-ray can cause the breakage, the break in the DNA. This one, the backbone of the DNA, which is the phosphate and sugar backbone of the DNA. Okay, when that happen, it can cause deletion. If this deletion is somehow occur on the gene, then you have a changes in the gene. Okay, but if it is occurring on region that do not have gene, then you can only have changes in terms of the number of repetitive sequence. So the fingerprinting is different, but the individual is normal. Okay, so that is the X-ray that can cause changes on the DNA. Okay, and then UV light, sometimes it can also produce a dimer. A dimer, you imagine when two adjacent thymine, they form additional bonding. Okay, usually this sugar phosphate backbone, you have another one over here, they only form a what bonding between this nitrogenous base and the other one. They form a hydrogen bonding, right? But with the two adjacent thymine, if you expose to UV light, sometimes they can form additional covalent bonding. Okay, not between this one and the other strand of the DNA, but on the same strand, they form additional covalent bonding, which is actually like this. Okay, you have methyl group over here, you have additional bonding over here. So this one will cause a dimer. So this is a dimer, and if it so happen that during this dimer, they become a not translation, a replication is occurring, the DNA cannot recognize this, so they will just add in random nucleotide. So the new strand will have a different sequence compared to the original one. Okay, because for example, if thymine dimer, if no dimer, this one will be AA over here, right? But when somehow there is a dimer and this one replication is occurring, A cannot be recognized, they cannot recognize the thymine, so polymerase cannot add adenine to pair with this one. Polymerase cannot add adenine, they will just randomly add in another other nucleotide, or sometimes they just skip this area. Okay, so it becomes shortened, deletion can occur as well. Okay, and then the next one, how mutation can occur is oxidation from free radicals formed by irradiation damages, uh, formed by irradiation which damage the individual bases. So free radicals occur in our cells all the time. Okay, Song, does this mutation affect individual or just next generation? Which mutation? You are talking about any of the mutation, isn't it? I'm assuming that you are talking about any mutation. As mentioned before, uh, how do you pass your gene to the next generation? Think about it for everyone, not only for those who ask questions. How do you pass your gene to the next generation? How does the plant pass gene to the next generation? How does the animal pass the gene to the next generation? And anyone might want to try type in the chat how you pass to the next generation before I answer. What is KH Song asked, which is a good question because you know I mentioned about this probably not that clear. Through gamete, yes. Okay. Yes, through sex chromosome. Not only sex chromosome, all chromosomes. Okay, but in the gamete during the I think you have an idea, which is when fertilization occur with the egg and ovum, isn't it? So to answer now, to answer what Thong asked, okay, you for uh, passing the mutated gene to the next generation, it has to go through this uh, through the formation of the mutation has to occur in the gametes, okay, through the gametes, in which during the formation of the ovum, during the formation of egg, okay, during the formation of sperm, if there are mutations that occur and it is somehow fertilized, then it will be passed to the next generation. Okay, but if I'll just like I mentioned just now, if you go to the tanning salon to have your skin tan, become brown like Donald Trump, then if you don't cover properly, then it can also expose those gamete cells, which then if mutation occur, it will be passed to the next generation when those cells are fertilized. But if somehow on your skin, then it might occur cancer on that area, so that one is not passed to the next generation. Okay, good. Okay, so that is how it is done. All right. Okay, and you have to remember that the mutation can occur not only when you are exposed, like you are exposed to the UV light. Okay, sometimes things that we do, we eat something when you know. Sometimes when we eat something, it doesn't happen, and nothing happened to us. Just like you, I'm quite sure you have heard people when the smoker, you know, the smoker keep on saying I've been smoking for 30 odd years. I'm fine. Okay, I'm fine. Nothing happened to me. All right. So I've been drinking for many years already. Nothing happened to me. That because you know smoking cause cancer, but why that person do not have a lung cancer, throat cancer, for example? It is because uh, different individuals have different uh, threshold to trigger something. Okay, for even so, even for our gene, for cancer gene, if it is present, if some triggering mechanism was not triggered, because the absence of these chemicals do not reach certain threshold, the individual will be fine. Okay, so different individual have different threshold, which then require different levels of mutagen in order to trigger the mechanism for the cancer. So that is why some people, even if they carry certain certain cancer gene, okay, they do not have cancer. Okay, so that is why those smokers, for example, be careful, stop smoking. Okay, all right, so free radicals, another one. Free radicals occur all the time due to environment, exposure, our exposure to the environment, and so on. So you can have active oxygen species in the cell, and this active oxygen species, or sometimes reactive oxygen species, if you read in a magazine or anything, reactive oxygen species can cause mutation, okay, in which it will have, for example, oxidation of this hydroxyl group, become O over here. And when this happens, this molecule will then bind with different codon. Okay, so oxidative damage of the guanine, for example, can cause guanine oxide. It will pair with different nucleotide when the replication occur, and then it will cause mutation to those cell. All right, so this can happen. So how do you avoid? One way is by taking this what those antioxidant. All right. Okay. The next slide is mistakes during replication alter genetic information. Okay, you remember this? Normal sequence, normal DNA strand, and then somehow oxidative damage occur to this strand, to this guanine, and then undergo replication. And this one as well, you can see replication. So bear in mind, when you have, or when the DNA has a mutation, the changes that is considered become a mutation, a mutant, is when it is passed to the next round of replication. It is preserved over there. Okay, so this one you can see from GC, the next 2 rounds of replication can have different sequence over there. So it is not when the mutation occur. Okay, for example, you take this specific strand, when G become GO over here, it's not like this C is kicked out and replaced with the A. Okay, it doesn't happen that way. Actually, this one, the C is maintained, the G become GO like this, and then you have to imagine this, when during replication, this one will become a temporarily single-stranded on both sides, isn't it? So the one with the GO, it will form, the GO will bind with A, it become a mutant. Okay, this one maintain as GO, it will become a mutant, but this one, it will have a normal sequence cell. So this is the one that go on, multiply again, it become a group of cells, a group of cells that have a mutant sequence. Okay? All right. Mistake during replication alter genetic information. As I mentioned, this is a polymerase, wrong base is added. Okay? And then you can see here 3', 5' exonuclease cuts here. Errors during replication are exceedingly rare, less than once in what? 10 to the power of 9 is what? 1 billion base pair. Okay, 1 billion base pair. So every 1 billion, you find only one mutation or error during replication. Why is that? Because of the process of repairing what you call proofreading is being done. So after proofreading, that is why every one mutation every 1 billion base pairs, which is quite rare, very rare. Okay, so why you got only one every 1 billion? Because proofreading enzyme make correction for the others. Okay, correct errors made during replication. You see here, DNA polymerase has 3' to 5' exonuclease activity, and this is not the only proofreading enzyme. Okay, so DNA polymerase, if you recall your genetic class, general genetics, different type of DNA polymerase, right? Some has several subunits. Okay, some are only polymerase that can do the same work of adding nucleotide, and at the same time it will reverse if it detect it adds in wrong nucleotide. Okay, so the one that go reverse, go in reverse to replace, remove and replace the wrong insertion. That one is the 3' to 5' exonuclease activity. Okay, why is it called 3' to 5' exonuclease activity? Exonuclease enzyme. Anyone would everyone can guess. Okay, everyone can guess. Someone who want to type, please do so. Why is it called 3' 5' exonuclease? Anyone would like to try? Why is it called so? Why is it so? Nobody try? Okay, good. Several of you have tried. Well done. Okay. Because it reverse, yes, it go back. Because the template is 3', 5', correct. It moves 3'. Well done, everyone. I'm quite sure everyone has that idea. Okay, because we look at this trend, you remember the DNA are synthesized from 5' going through 3'. Yes, it is because so when it goes that way, meaning to say it goes from 3' going back towards the 5'. So that is why it moves to the 3', 5' direction. Okay, well done, everyone. So, and then the enzyme is exonuclease because it can recognize the terminal end, the outside of the nucleotide. So exonuclease. But somehow if there is another enzyme which can bind, okay, and then it can chew or cut the region over here and it can move backwards, then you can have 3', 5' endonuclease. Okay, but if there's another enzyme that come in the middle, remove this and move forward, then you have 5', 3' endonuclease. Okay, so that is how different proofreading enzymes are done. So usually, so for this one, the idea is that polymerase can do proofreading, and one of the example how it do it is by having the 3', 5' exonuclease activity. Okay, so that is why we have a very low number of mutation. So this one every 1 billion means that somehow even polymerase didn't detect the mutation. Okay. Unequal crossing over creates one homologous chromosome with a duplication and the other with a deletion. So you remember, mutation not only due to changes in the nucleotide, but can also have this homologous, you can have homologs that can cross over, unequal crossing over, which means that one of the chromosome move to the other side. Okay, so you can have unequal crossing over, like mentioned this one, homologous chromosome, red photoreceptor. This one is an example. Okay, it can occur for any other gene as well. So in this case, they investigated homologous chromosome, okay, chromosome pair in a diploid for red and green gene, and then somehow you can have unequal uh, pairing in which the gene, because they are quite similar, sometimes the green uh, can align with the red one, and crossing over can occur. Okay, when crossing over occur at this region, so you can have a deletion on one side and duplication on the other side, although they are a homologous chromosome. All right, so unequal crossing over. You can have a transposable element in certain plant. For example, you can have a mechanism in which they can move from one chromosome to another place. Okay, if you recall the study by Barbara McClintock. Okay, so she discovered uh, transposition, yeah, in which the chromosome fragment or segment can move to another chromosome. Okay, so that one is a transposable element, and there are mechanisms on it in order for it to go from one place to another without needing additional molecule from the outside. Okay, so that's why a generic term or layman's term for transposable element is a jumping gene. Okay, mutation increase mutation rate using different mechanism. Okay, so base replacement. Okay, and then you have alteration of the base structure. Okay, and properties. For example, due to certain agent. Okay, for example, like this one, base analog. Base analog is a chemical molecule that behave like a nitrogenous base. So that is why it's called analog. It's not like an analog clock, automatic clock, okay? But analog, which means that they have similar properties, but they are different molecules, okay? Base analog have a chemical structure almost identical to DNA nitrogenous base, bromouracil, BU, okay? Bromouracil is almost identical to thiamine. Okay, thiamine, which then can pair with adenine. So, and bromouracil. Okay, if you might ask, if bromouracil, the BU, behave like a thymine, so in the nucleotide, it will pair with adenine. So adenine, when it pair next round, it pair with bromouracil with bromouracil, then in the other round of replication, it will always pair with adenine. So there's no changes, right? But what happened is that this bromouracil, although chemically it is behaving like thiamine, sometimes in different condition, you call it rare state. Okay, you just imagine like a person when in a moody state, everything is not correct, everything is wrong, in a moody state. Okay, so in a rest state, it will pair with guanine. So that is why the same bromouracil in a normal condition, it is pairing with adenine, but in the replication somehow, during that replication, it become in a moody state. Okay, it become moody. In the rest state, it will then pair with guanine. So guanine is different from adenine. So you can see from the TA, it will move to CG. Okay, so this analog can cause mutation. The other one is a hydroxylating agent, in which the name itself suggests that it is the one that add a hydroxyl. Hydroxyl is a OH group. Okay, hydroxylamine add OH to the cytosine, And with the OH, the hydroxylated C now pair with adenine. So it behaves differently. So cytosine supposed to pair with guanine, but when it is hydroxylated, this hydroxylated group will then allow this to pair with adenine. Okay, so you can have from CG become a TA mutation. Okay, so okay, CG become TA. What is it? What is this? Is it transition or transversion? Okay, what is this? CG become TA. Is it a transition or is it a transversion? Anyone can try? Uh, it's not transition. Well done. Okay, it is a transition. Yes, transition because guanine changed to adenine. This one is a purine. Guanine is a purine. Adenine is a purine, right? So purine to purine is a transition. Well done, everyone. Okay, and then alkylating agent similar in concept like this. Okay, and then deaminating. So the word deaminating straight away, what should come to your mind? Amine. Okay, amine means what? It should have an NH. Okay, amino acid because it is called amino acid because it has NH. So amine group, it has NH. So deaminating agent, meaning to say the removal. Okay, remove amine group. Deaminating agent, remove amine group. The concept, all the concept, whether it's a alkylating, or whether it's a hydroxylating, it change the behavior of the nitrogenous base. Okay. Okay, and then so that is similar concept like the one before. Okay, another method of how mutation can be done is by insertion between bases by intercalating agent. So this one, if you remember, this one is the double-stranded DNA double helix, isn't it? So you can have what you call this region, this region, this region, this region. What do you call that? Anyone can recall? Start with a G. It has a big and small G. Okay.
Is it groove?
Yes, correct. Well done. Okay, it is groove. So major and minor groove. So it's not like it is a solid like this. You remember they are coiling. So just like you imagine spring, but in a double helix, so there are region whereby you can insert something, right? If you have a helix, I don't have any example. If you have a helix, you can insert something in between. Okay, when you insert something, you are doing something what you call intercalating. So you insert something inside there. So proflavin intercalates into a double helix at the groove area, which then disrupt the DNA metabolism, eventually resulting deletion or addition of the base pair. So it just insert itself, and when it insert itself, you imagine this one will create tension. Okay, if intercalating agent comes over here, it will then cause tension over here. When that happens, and at the same time, it is undergoing replication or recombination, then you can have random insertion can occur, or sometimes it causes deletion in the area, okay, because the enzyme found that this region got a problem. I don't want to deal with it. I just remove this one and proceed with the next. So you can have deletion as well. So you can have insertion or you can have a deletion due to the presence of intercalating agent. Okay, so next semester, for example, you will be going, if you come back to campus in the second semester, if you go to the lab, then you will have STB2163, and one of the compound that we use in the lab is the ethidium bromide. In order for you to look at the DNA, it will also cause intercalating. It is also an intercalating agent which can cause mutation if you are not careful. Okay. All right. Repair enzymes fix errors created by mutation, similar in concept. Okay, similar in concept, in which there is, if there is a mutation. Okay, I'm going to do this very fast because I plan to do it 1 hour only, so that your time, you don't spend too much on your data. Okay, when you have a mutation, for example, this one causing dimer. When you have dimer, the enzyme recognize, it can identify, it can cut the region and replace with polymerase, can come and replace with the correct enzyme, as correct nucleotide that do not have the dimer over here, and then the final one between the adjacent nucleotide, the phosphodiester, because thymine, because for example, like this one, G and C is present. Okay, but then the final phosphate sugar connection is not done, so ligase will come and do the connection. Okay, so ligase is still playing a role in addition to when you like you learn in replication and so on. This one can also play a role. So repair enzyme fix the errors. So this one is additional enzyme that is not done, the repair, when the repair is not done by the polymerase enzyme. Okay. So, okay, okay, direct detection. Hold on, yeah. I want to find out how many slides you have to go. Oops. We are only halfway through. Okay, there's still a plenty. It's already 1 hour. I was quite slow today, isn't it? Okay, shall we continue next week? Shall we continue next week? Or you don't mind to carry on for another 1 hour? Okay, we take a break. I'll give you 3 minutes to decide. You can type in the chat, and based on the majority, then I will whether proceed or stop. Okay, because you know, I can understand some of you wouldn't want to be online for a very long time due to several reasons. Okay, I can understand that. Don't worry. Okay, we take let's say 1 minute break. 10:03. I come back and 10:04. Okay, you can chat up to me. Proceed. Okay, okay, some of you say that I can proceed. It's up to me. Majority wouldn't mind if I proceed. Okay, then, okay, thank you everyone. But so for those who are unable to join, don't worry. Okay, don't worry about that. You can always, I will upload the YouTube video link into the Elite. You can always continue your continue watching the lecture based on the YouTube. Okay, thank you everyone. So now we continue. All right, the direct detection of genotype distinguishes individual genomes. Okay, all right. I have mentioned just now that mutation. Okay, mutation can occur. Okay, in various places on the chromosome. All right. You have to imagine that when you are exposed to X-ray, when you are exposed to mutagen and so on, what happens is that most of the time it is not targeted. Okay, it is not like when you expose to UVA, it will target polymerase gene only. Or when you expose to UVC, it will target hydrolysis gene only. Okay, it is not that way, but it is randomly occurring. So if the mutagen somehow found or uh, change the sequence on the DNA that happen to be gene, then if it is on the uh, gamete, then it will be passed to the next generation. But if the change occur in the gamete but not on the gene, changes can occur, but that changes only change in terms of the DNA that is not on the gene. So the individual will be normal because the gene are not affected. Okay, so so mutation can occur anywhere on the DNA on the chromosome. Okay, if it happen to be on the gene, then it will change the individual, and if it happen to be on the gene and the cell is the gametic cell, it will be passed to the next generation. All right, so it can occur anywhere, and basically you can see the difference when we can detect it basically. So direct detection of genotype distinguishes individual genome. So changes that occur on us, okay, whether through mutation, and you have to bear in mind, okay, changes that occur on individual is through mutation, and that other type of changes that can occur on our DNA is due to recombination between the homologous chromosome inside us, as well as when the fertilization occur, then you can have different combination. So that is why you and your sister, you and your brother are different. Okay, because changes that occur can cause variation among individuals, and we can detect it. Okay, so individual genomes means that you can see different individuals, so you do not need to do a genome sequencing to see the differences between individuals. Okay, members of the same species show enormous sequence variation in the genome. So this one, you imagine, just a simple one is if you are not identical twin. If you are identical twin, that one is different story. Okay, but if you and your brother or sister, although in terms of the genes we have the same gene, so that we are normal, but in terms of the DNA sequence, we are slightly different. So 1 in 1,000 base pair differ in any two randomly chosen humans. So we have always different DNA sequence, okay? And most of the time in normal individual, the difference in the DNA sequence is because of changes that occur on a non-gene region, on the region that is not having any gene on it. Okay, so the difference is present, but not on the coding of the gene itself. Since the haploid genome is 3 times 10 to the power of 9 base pair, this one is a human genome size. 3 times 10 to the power of 9 is what? What do you call it? 3 billion base
3
pair.
billion
Okay.
base pair.
Yes, 3 billion. Yeah, 3 billion. 1,000 million is 1 billion, right? So 310 to the power of 9 is 3,000 million, which is 3 billion. So human genome in a haploid form is 3 billion base pair. So 1 in 1,000, meaning to say human genome 3 billion. So we have all individuals mostly have 3 million differences in terms of the DNA between us. Okay, you, your friend, between you and me, and so on. We have 3 million differences. So you can use this when you talk, you know, with your friends, that especially who do not sit for this class lah. Okay, you can say that, you know, between you and me, there are 3 billion differences between us. Okay, you can say that. And your friend must be wondering why is that so. Okay, there are 3 billion 3 million differences between you and me. Okay, 2 randomly chosen, so anyone. And most, as I mentioned before, most differences are in a non-coding, non-regulatory region. So that is why changes can occur, mutation can occur in us, okay? And that mutation do not affect us because it is occurring on the non-gene or non-coding region. Alleles, any variation in the genome at a particular location. Okay, but this one, when you talk about allele, we have 2 copies, right? We have 2 copies or 2 homologs, chromosome number 1, got 1 pair, chromosome 2, 1 pair, 3, 1 pair, and so on. Okay. And the allele, if you still remember, in the Mendelian genetic, the gene that code for color. Okay, if big A for red color, when big A pair with a small a, it will produce red color only. Small a, white color. So that A gene actually located on the same position. Okay, homologous chromosome. If they are on the same position, okay, because this one is black and red, this one is a what? What do you call it? If this one is, let's say this one is black and black, what do you call? Black and black. Anyone would like to try? If black and black, what do you call? Same position, black and black. Nobody has tried yet. What do you call it? If on the chromosome and the region with a black and black. Okay, only Nabila has tried. Well done. Narisa, homo something, correct. Homozygous. Okay, homozygous. Okay, well done. Those who remember, well done. Those who have an idea, something you kind of know but you cannot recall, okay, try to do revision. Is a homozygous. But if it's black and red like this, it's a heterozygous, right? So, and this is called an allele. So remember, an allele not necessarily be referring to a gene as well. Okay, for example, this is a gene coding region for a gene. This region is not a gene, okay, but because they are homologous, there might be some differences between this. So this one is also an allele for this region. Okay, so you can have some changes, some differences between this. So heterozygous for this allele, for example. So alleles are any variation genome that at a particular location. So at a particular location, if there are differences, then that one is called allele. So location is locus, locus. Okay, so it's quite easy for you to remember, isn't it? Locus, you relate to position, location. Okay, polymorphic, 2 or more alleles at a given locus. Okay, polymorphic, 2 or more alleles. And polymorphism, the particular variation. For example, you have 3 copies at this region, 3 repetition, but because this one come from the mother, this one come from the father, got 2 repetition only at this allele. Okay, so this one is a polymorphism. So because this one is 2 or 3, then polymorphic. All right. So, and this region that has polymorphism, you can use this as a marker to do a study to see differences between different individual. Okay, so this one in you got 2 or 3, and some other individual 2 and 2, another individual 3 and 4, for example. So this particular region can be used as a marker. All right. Okay, and sometimes for a locus, you don't know what the function is. Position of the on the genome with no known function. Okay, so that is how it can occur. So polymorphism, polymorphic, different allele changes to them can occur because of mutation by changes in on the DNA, or sometimes mutation by changes on the chromosome segment. Okay, so when that happen, you can have polymorphism. So if you recall back from our first lecture, you can have genes, okay, and genes can have series. So that one is different topic. Okay, and then changes to the gene can cause non-function. So because of mutation. Okay, so mutation can cause changes to the gene, and mutation can now also not cause changes in the gene, so the individual will be the same, but mutation cause changes in the DNA sequence that differentiate between one individual to another. Okay, so when that happen, you have this what you call polymorphism. So changes that is differentiating between different individuals. Okay, and changes can be big or small. When the difference between individual are very small, you can have SNP, single nucleotide polymorphism. So big, small differences, and then slightly bigger, you go down to microsatellite, mini satellite, and then DNA segments, for example. Okay, you can have microsatellite, mini satellite, and then sometimes very small changes in between, not to the number of you call microsatellite. So you can have in between single nucleotide polymorphism and microsatellite, you can have small changes in the by deletion or duplication. Okay, but normally when we talk about polymorphism, we are talking about these three, whether it's a SNP, microsatellite, or multilocus. And SNP, the way it's pronounced is not SNP. SNP is pronounced as SNP. Okay, SNIP, SNP, SNIP. SNP equivalent to single nucleotide polymorphism. Okay, so now you see that 3 million differences between you and me, okay, or between you and your friend. We have 3 million differences between us. That 3 million can be because of this single nucleotide polymorphism or SNP on some area, microsatellite differences on some area, and mini satellite differences on some other area in our chromosome. Okay, so the size is 1 base pair. Okay, number of allele is only 2. Okay, whether it's a AT or GC, isn't it? Okay, and then microsatellite, you can have different alleles, 2 to 10, meaning to say that if we have for SNP, okay, sometimes between there are 10 individuals in the room, we can only have 2 different types of differences between 10 of us. Okay, whereas in microsatellite, the number of alleles, we can have each of 10 of us can have different arrangement for this region, for example. Okay, even for many satellite like this. Okay, so number of population, meaning people affected and so on. So usually for DNA fingerprinting, we use a mini satellite. For linkage mapping, to see the connection between For example, in mapping of the restriction enzyme, for example, linkage mapping, gene mapping. So usually we use at the microsatellite and mini satellite. Microsatellite and SNP level. Okay, and then how we detect by PCR most of the time. Okay, bigger DNA. This one, PCR at this mini satellite. Why we don't use a PCR? Okay, hang on here. I'm going to answer the phone. Okay, sorry for the interruption. Okay, for mini satellite, we don't use PCR because the DNA is very long. Okay, very long. If we do a PCR for the mini satellite, actually we can, but it's just a waste of consumables lah, waste of chemicals, waste of everything. So we just do a southern hybridization. It's much easier in a way as well. Okay, so that is how we differentiate between individuals. SNP is a single base pair substitution arise by mutagenic chemicals or mistake in the replication. Only two alleles. Okay, only two alleles. Whether it is a AT, the other one is a GC. Okay, ratio of alleles ranges from 1 in 100 or 50-50. Sometimes these are the ratio. Okay, and then 20 years ago, 5 million human SNPs have been identified. Most occur at anonymous loci. What does it mean? This one, the differences between individuals for SNP in the region that do not code for any gene. Okay, anonymous loci, or do not code for an region that we don't know what the function is, unknown region. Okay, so that one is anonymous loci. Mutation rate, okay, 1 for every 1 billion per generation. Very few. Yes, anything? Any? Okay, any question? Okay, useful for DNA markers. All right, you have to remember SNP, mini satellite, microsatellite, not only being used in human, but for other organism as well. Okay, Dr. Ho, for example, use SNP to identify the kelampayan. Okay, can everyone hear me?
Yes.
Yes, we
Can,
can,
yeah?
Doctor.
Okay, at the moment, some of you mentioned cannot hear. If you cannot, if other can and you cannot, probably on your side, you try to exit and re-enter again, yeah? Try that first. Okay, SNP can also be used for other organism and not only human. Okay, for example, as I mentioned, our study on sago plant, we find SNP. Okay, previously Dr. Ho, he used SNP to see the differences between timber plant, timber tree, kelampayan that is superior and the normal one. So it can be used in many different organisms. It can be used for fungal study as well and so on. Okay, microsatellite is slightly bigger as mentioned before. One every 30,000 base pair, not necessarily exactly one every 30,000. Okay, repeated units can be 2 to 5 base pair in length. What does this mean? So you imagine you have only a sequence of C and A like this one. So this one is a repetitive sequence of CA. So you can see here CA in a parenthesis 15. So 15 repetitive sequence of CA. So this one is a microsatellite. Okay, sometimes you got CAGA. Okay, CAGA 4. Okay, 4 repetitive sequence of CAGA. So it repeats 10 times, for example. So repeated units of 2 to 5 base pair, and it can go until very long. Okay, this one is mutated by replication error during the replication, and mutation rate of 1 for every 1,000 per locus per gamete. So microsatellite can also be used for a polymorphic DNA marker. For example, if I want to differentiate between our study on pineapple, we find MD2 pineapple compared to Josephine pineapple. Okay, the repetitive sequence of, for example, like in this case, the CA, this one is 15 times. In the Josephine is 30 times. So you know that if I want to find out in a tissue culture whether this one is a Josephine or whether this one is MD2, I can use a PCR. If I find like if 15 times 2, right, 30 base pair, then this one is MD2. But when I do another PCR using the same primer, it find, for example, if this one is 30, 30 times 2, 60. So 30 versus 60. So I can determine which one is MD2, which one is Josephine. So you can use as a marker. Okay. So PCR to rapidly isolate and amplify DNA target. Okay, I am going to skip the PCR. Okay, not skip, just go very fast. Okay, for example, if in the microsatellite, I want to do a PCR to determine between MD2 and Josephine pineapple, right? So the region is the one that is going to be amplified for the repetitive sequence of the CA. So this is the region. In the MD2, in the Josephine, they will be the same region, but in MD2, it will be repetitive 15 times repetition of the CA, whereas in the Josephine is 30 times. So target region is the same, beginning is the same, ending is the same. So the beginning is the primer, the ending is also the primer. So we have forward primer or left primer, reverse primer is the right primer. Okay. So for the two different pineapples, I will have the same primer, and then I do an amplification. So when I do amplification from one copy, okay, let's say if this one copy, I can copy it many rounds. At the end, I will get 1 million after 30 rounds. Okay, because this one is exponential. So in the PCR, you got 3 cycles, for example, the one at the 94 degrees. Okay, and then the one at 50 to 60 degrees, and another one is 72 degrees. Okay, so you can have 3 different stages in one cycle. So it usually repeated 30 times or 30 cycles. Okay, so you can have this one is annealing, this one is amplification, this one is the what do you call separation. Okay, so 94, 50 to 60, and 70. So you can have like this from one copy. You can have 30 cycles. At the end, you can have 1 million like this. From one copy originally, from 2. 2 become a template, become 4. So 1, 2, 4, 8, and so on. So after this one cycle, 2 cycle, 3 cycle, 4 cycle, 5, 6. Until 30 cycles, you can have 1 million. Okay. So those amplification, why do you do that? Because you want to amplify the same thing so that you can visualize under the using the gel electrophoresis. Okay, so the function of the PCR is to multiply or make a copy of the same area many times, usually 1 million times by the end of 30 cycles. So that's the reason why we do PCR. No, because sometimes we get a sample very small. We want to detect, you do not have that sample anymore. Okay, for example, from the crime scene, you want to amplify target region to see how many repetitive sequence this region has. So that sample, that's the only sample you have. So you cannot do much analysis on it unless you amplify. So if you have very small amount, you amplify after 30 cycles from 1, you got 1 million in the end. So you can do analysis properly. Okay, so that is why you do PCR. Okay, so so PCR can be done to detect differences between individuals. Okay, for example, in this case, I am using this one. Okay, I just go straight to this microsatellite alert detection analysis of size differences. You have, okay, remember the one that I show example. Okay, allele. Different individuals have different allele combination. Some individual have 1 and 2, 1 and 3, 2 and 3, or 3 and 3, 1 and 1, 2 and 2, and 3 and 3 as well. Okay, different combination. So microsatellite can analysis will be able to tell whether you are homozygote or heterozygote. Homozygote means what? Whether you are having allele of 1 and 1, 1 and 1, which means that they are same size, or you are 2 and 2, or you are 3 and 3. So you have homozygote of 1 and 1, 2 and 2, or 3 and 3. And in some individual, they have a combination of 1 and 2, heterozygote. Okay, meaning to say in your chromosome, you have allele 1 and allele 2. So you are a heterozygote. And in some individual, you have 1 and 3. And in some other individuals, you are 2 and 3. So you do a PCR, you can amplify, and then from the 6 samples that you have, you run on the gel, you can see differences like this. So you can determine the individual. Okay, so this one is how you detect the differences in individual. So what is the PCR got to do with this? PCR is just to amplify, to make it many copies, so that when we run, we can visualize it using the electrophoresis. Okay, so determine sequence flanking microarray. So coming back to this, okay, you have an individual. For example, individual with allele 1 and allele 2. When you have PCR, you will amplify this. You will amplify this. You will have amplification of the allele 1, many copies. Because this one is short, when you do a gel electrophoresis, it will move very fast. It go down to here. This one is slightly longer. When you do a PCR, you can get many copies. When you run on the electrophoresis, all of them will be at this position because they are the same length. They are over here. Okay. Okay, one question for you. Can I do electrophoresis with only this? I straight away go to this. Can I do that? Or can you do that? From here, I straight away go here. Can you do that? And then see this. You skip the PCR. Anyone can try? I think you okay. No, cannot, no, cannot, no. Okay. All right. Actually, you can. You can do the electrophoresis, but you cannot see. Okay, you cannot see because it's only one over here. Very small amount, very little amount. you cannot see, but you can do electrophoresis. Some of you answered, you can only produce. You can do this, but you cannot see. Okay, so you can take this and run, but you cannot see it. So that is why we do a PCR, so that this one can have 1 million, this one can have 1 million. Only with 1 million we can observe under the electrophoresis. Okay. For example, Amalin say produce same result. Yes, theoretically they are at the same position. Okay, they are at the same position because this one and these are a copy of this, so they will migrate to the same position. But if you do this straight away, you cannot see them. So that's why you do a PCR. Okay, so if many of you answer no, I accept that as a no, because no means that you cannot see. Okay, but if some of you answer yes, yes means that you can do this. It is actually there, but you cannot see. Okay, so microsatellite can be used to detect the polymorphism. So in some other individuals, you have homozygous. Some of them as heterozygous. Okay, gel electrophoresis is just preparing. I'm going to skip this one. I think you can watch a video on the electrophoresis. So, and then the next one is deletion, duplication, and insertion of the DNA. Expand or contract the length of non-repetitive DNA. So you can see this one, between the difference of allele1 and allele3, because when the chromosome undergo replication, sometimes in the parent, okay, let's say this one comes from the father, this one comes from the mother. During replication in the mother, in the gamete cell, it has deletion. So some of the CA are removed, become short. In the other parent, during replication, some are inserted, so they become longer. So deletion, duplication, and insertion will expand or contract of this repetitive DNA. Okay, and then crossing over can also cause deletion, duplication, and then insertion by transposable element can also cause deletion and insertion. Okay, and then much less common than other type of polymorphism. So less common for this one to cause polymorphism. Okay. Uh, okay. 1037. Fragmenting complex genomes into pieces. Okay. Hold on. I think producing results. Sorry, may I ask how to see the difference in the diagram for 1.3 and 2.3? Referring to, I believe the question is referring to this one, correct? Okay. May I ask? Okay, may I ask how to see the differences in the diagram for 1.3 and 2.3? Okay, 1.3, if let's say, if there are individuals, okay, because this one is example 1 allele 1 allele 2. Actually, in another individual, there is, for example, individual with 1 and 3 over here. So 3 is different from 2 because of the number for 3 is higher than number 2, right? So when 3 is bigger than 2, when you put on the electrophoresis, it will migrate slower. So that is why 3 is over here. Whereas number 2 moves slightly slower. So that is why it is over here. So when you have an individual, okay, for example, 2 different individuals, one with a 1.3, another individuals with 2.3. Okay, so with a 1.3, when you run on the electrophoresis, 1.3 meaning to say one of them will move very fast. The other 1.3 will move slow, quite slow. It will when you stop, it is this area. But for the other individual, because that individual has 2 and 3, it will move slightly slower than 1. So after the same time, okay, for example, if these are run after 1 hour, after 1 hour, you stop. So after 1 hour, number 1 will be positioned down here. After 1 hour, number 2 will be positioned over here, and number 3 will be positioned over here. So that is how you differentiate between allele 1 and allele 2. Because allele 3, they are very long, they move very slow. So for the concept of electrophoresis in this case, you use the same length of time to allow for them to move, because you start from the top over here. Okay, in the electrophoresis, you start from the top. You start from the top, and then your DNA will move down after 1 hour. Because this one is down here because it moves very fast, because it is short. This one's medium speed because it is a medium size. This one is a very long, it's a very slow speed. Okay, so that's why you differentiate. You run them for the same time, same length of time. So if this one is very short, this one is very slow, very short, very slow. The short one will start, but it will move very fast. This one moves very slow. So after 1 hour, it stops over here. So this one is short, it will be positioned down here. This one slightly, because it's slightly longer, it will move very slow. After 1 hour, it will stop over here. Okay, so that's just like a rabbit and turtle. When they do a race, this one very fast, this one very slow. Okay, so that's you differentiate. They will show differences after 1 hour. Okay, so that's how you okay, and then another question. If the electrophoresis show only 1 bar, it means that the individual have the same allele. Correct. Okay. For example, like 11, 22, 33. They, because 11, the other allele also 1, meaning to say the same size, right? So when the same size, when you mix together, you just base according to the length, they will move the same. So although they are and they are the same allele, so they will move at the same position. Okay, so that is why you see only 1. So that is why you see only 1, because okay, these two, right? This one from the father, this one from the mother, same size. But when you are in the electrophoresis, how many can you see? You can see only 1, isn't it? Although there are 2, but you can see only 1. Okay, because they are the same length, same as this one. Okay, you only see 1, not only because it's 1, but it is only 2, but they move at the same time because they are same size. Okay, may I ask why allele 3 move at a shorter distance as opposed to allele 1 that move far? Okay, you imagine okay, electrophoresis. I try to. Okay, electrophoresis consists of gel. Okay, in which if you imagine this one just like a maze, in the movie Maze Runner, right? Maze Runner movie. You can go from in a Maze Runner movie. You can go from one end to another end. Many ways you can be done. So the one that is the shortest one, if you can find a very short distance. Okay. The longer you can, sometimes you will move very different way, zigzagging, and you reach here as well. But at the same, but at a longer time, right? So if 1 hour, the shortest distance will be until here. In 1 hour, if you move zigzagging because of the obstacle, in 1 hour you only reach until here. So after 1 hour, you arrive only here. Okay, so for the electrophoresis, it's just like the DNA is moving through a series of holes or obstacles. So these are all obstacles, but the DNA with a very long DNA is, you imagine it as a very big person. So very big person, to move through the obstacle, it takes some time. But for a very small person, to move through this obstacle, it just wiggle through, just very, very fast. It move from one position, from here until here, very fast. So it reach here. But for a very long molecule, it has to move through this obstacle, very, very difficult. So that's why it is slow. So that is try to explain what Risa asked. Why allele3 move shorter distance opposed to allele1? Because allele3 has to wiggle through, because it is very long, it need to wiggle very, very slow. Whereas allele1 is very short, it just go very fast. Okay, so that's why within 1 hour, this one has reached over here, but number 3 is still up there. Number 2 go midway because it's slightly shorter, so slightly faster than number 3. All right. Okay, so that is how we differentiate the length of DNA using the electrophoresis. Protein concept is also the same, yeah, but you will learn about this later in other course. Okay, fragmenting complex genomes into pieces for analysis. Okay, sometimes on the DNA, very long DNA, you cannot analyze very long. So you need to make it very short, and you can cut the DNA into shorter pieces because on the DNA got a restriction enzyme. Okay, so you can fragment the long DNA in the DNA, in the genome, into smaller pieces because on the DNA sequence there are restriction enzymes, and restriction enzymes can recognize specific places based on the nucleotide arrangement so that it will only cut at that particular position. Okay, for example, this restriction enzyme for TAC1, it will recognize TGCA. If TCCA, no, TCGA, I think this one. TCGA, it will only recognize TGCA. It will not recognize other sequence to cut. Okay, so if it come across TGCA, it will cut. For RSA1, it will, when the enzyme scan through the DNA, it found, for example, GTAC. If this GTAC enzyme recognize, it will cut the DNA into 2 pieces. Okay, for EcoR1, and so these are different enzymes. Yeah, so if EcoR1 enzyme scan on the DNA and then it find, oh, GAATTC is over here, then it will cut. It will cut the DNA. Okay, so that is how the restriction enzyme function. So different enzyme recognize different sequence of the nucleotide so that it will cut on the DNA. So if I have 2 enzymes, for example, if I mix EcoR1 with BAMH1, okay, if there are one region, okay, okay, I annotate. Okay, for example, yellow color, not nice. Red color. Okay, this one is BAMH1. BAMH1. On the region, this one is EcoR1. Okay. If I use one enzyme, I hope you can see, yeah. If I use one enzyme, BAMH1 only, on this long DNA, it will only cut at this position. In another tube, the same DNA, if I use EcoR1, it will cut only at this this one will be left out because different sequence, yeah. But if I mix BAM H1 and Eco R1, for example, I mix together these two enzymes and I put in the tube of the DNA, they will cut at two places because BAM H1 will cut here, Eco R1 will cut here. Okay, because they recognize different places. Okay, so that is how you fragment. So in a long genome, okay, for example, very long DNA, okay, okay, these are the restriction enzyme for KPN1. So if I put this KPN1 for this DNA, it will cut at 1, 2, 3, 4, 5 places. So I will have 5 fragments. 2 fragment, 3, 4, and then this one lah. Okay, and then the other. So you get fragment, fragment, fragment like that. So that is how you cut the long DNA into smaller pieces. Okay, so that is how you do it. All right. Okay, I'm going to stop in 5 more minutes. So restriction enzyme can be used because different individuals sometimes at the same position, it do not have restriction enzyme. So when you cut into one individual, it has, another individual do not have. It will show like this. Okay, for this one, probably what I want you to do is that you go back and try to understand this, and I will explain further oh, this is the last one. Okay, lucky you. Okay, you try to figure out what is meant by this. Okay, the clue here is that